Welcome, everyone. Welcome to the Mid-Atlantic Micro-Nano Alliance virtual seminar series that we have going on this summer. Today we have a very special guest, and I'm here virtually with co-president Ryan Sokol. So Ryan, who do we have today?
So hello, everyone. Today we have Ashley Chapin, who's going to be presenting our MAMNA seminar. And so Ashley is a bioengineering Ph.D. candidate working under the advisement of Professor Reza Ghodssi and Professor William Bentley at the University of Maryland, College Park. Ashley received her bachelor's degree in biological engineering from MIT. And recently at UMD, Ashley was selected as a campus-wide winner of the 3-minute thesis competition. So Ashley, I'll let you take it from here.
Great. Thanks so much, Ryan and Heidi, for your help. Hello, everyone. I'm Ashley Chapin. I'm a PhD student at University of Maryland. I'm in the MEMS Sensors and Actuators Lab, or MSAL. And today I'm going to talk to you about my presentation, which is titled Hybrid Platform Development for In Situ or On-Site Monitoring of the Gut-Brain Axis. So most of us have an intuition about the connection between the gut and the brain, perhaps in the form of butterflies in your stomach when you're nervous. So this bidirectional talk between the gut and the brain is termed the gut-brain axis, mainly mediated by the vagus nerve, which is capable of sensing your gut environment and regulating parasympathetic adaptive responses in the brain. Think rest and digest, as well as your mood, your emotional responses, hormone control, sleep, and many vital behaviors. We know that a healthy diet makes a healthy gut, but are you aware that the gut is also home to a huge variety of gut bacteria termed the microbiome? These are the first line in defense against pathogens and are essential for digesting and allowing us to absorb the nutrients from our food. However, with increased use of broad spectrum antibiotics and even eating a Western diet, which is high in saturated fats and simple carbs, this decreases the diversity of our microbiome to become unbalanced, what we call dysbiosis. This not only leads to a number of painful and disruptive GI tract disorders, but these are also associated with many neurological disorders, such as depression, anxiety, Alzheimer's, the list goes on. Let me give one example of this. In patients with major depressive disorder, bacterial species called KLE1738 and Bacteroides spagilis are more abundant in their guts. And this was associated with impaired connectivity in the left dorsolateral prefrontal cortex. Lower activity in this area of the brain is associated with depression. So we have a strong clinical correlation between these gut and brain disorders, but we cannot truly claim that the bacteria cause depression in this case because we do not have a system to measure the specific impact of these bugs on gut-brain axis nervous signaling pathways. We also lack easy to measure biomarkers of both the GI and neurological disorders, which both of these conditions are mainly diagnosed currently based on symptoms alone. So getting to understand the system better would improve diagnosis of these disorders, but it would also help us to have a better framework to understand how health and nutrition impact everyone and their mental health on a daily basis. So let's dive into what we know about the gut-brain axis pathway. The enteric nervous system, or ENS, also termed the second brain, is key, which sends nerve endings throughout your entire GI tract. These ENS nerves are responsive to chemical communication systems that are built into the gut lining, or the gut epithelium that separates the lumen of the gut from the internal tissue and the enteric nerves. Enterochromaffin cells, or ECCs, are the main chemosensor in the gut. They're capable of sensing mechanical stretching or distension, such as from muscle contractions. They also sense chemical stimulants in your diet, like wasabi, for instance, and also changes in water content. They also respond directly to metabolites from your gut microbiome. For example, when you eat fiber in your diet, bacteria break this down into short-chain fatty acids, and these directly stimulate EC cells to secrete serotonin from their bottom or basolateral side of the cell. Now, serotonin is most well known as a neurotransmitter involved with mood in the brain. But actually, 95% of your body's serotonin is produced in the gut and mainly by these cells. Here, among other paracrine functions, serotonin stimulates enteric nerves and communicates these gut conditions to the vagus nerve and then to the brain. Currently, research is more familiar with the types of bacteria in your gut and their metabolites, and also how the vagus nerve sends signals around your body, but we don't have a clear picture of this serotonin release and signaling, nor do we understand how different bacteria or different diet components can affect this. And we also don't know how it specifically affects downstream pathways to result in different conditions in the brain. So the main reason that we don't have this information right now is that we lack the technology that can access inside this tissue to measure the molecules that are being released in real time, so that we can correlate that with the conditions that it's correlated with. So now let me give you a little bit of an overview of how we've come to know what we know about the gut-brain axis. And most of that is from animal studies, especially in rodents, and especially in germ-free mice. So these are mice that are raised in a sterile environment so that they lack any microbiome. That way, researchers can investigate the impacts on physiology and neurological development when an individual does not have any bacteria. And from there, if you have a completely clean gut, then you can infect that gut with any particular species of bacteria to see exactly how that affects the brain or development. So one example of this, researchers can assess anxiety-like behavior in mice using things like a forced swim test. And they find that this type of behavior is enhanced under increased inflammatory states. And that is caused by a pathogen infection, or even with diets of high saturated fats and high carbohydrates. So interestingly, when you perform a fecal transplant from a healthy mouse, which has a normal diverse microbiome, or even from a human that has different kinds of bacteria, and you transplant that fecal matter into a sick mouse who has a dysbiotic microbiome, this can reduce that mouse's inflammation and neural symptoms. It reduces their anxiety. It reduces the problems that they're experiencing. It sort of shows like there's a causative role in that gut microbiome. So these strides are very important because it's highlighted the importance of the gut microbiome in our overall health. But there are issues with animal studies, even beyond their obvious ethical issues. For instance, it is difficult to compare results in rodents to actual human physiology. And it's really hard to isolate the actual causative factors in these whole organisms. Where there's so many factors going around, it's hard to identify how the signal is actually getting from one place to another. So instead, what we can do is grow human cells in engineered platforms, which we call gut on a chip, to study specific functions such as nutrient absorption, for example. So just like electronic chips in your phone, engineering techniques can be applied when designing these gut chips, such as including vacuum chambers to stretch the gut layer to stimulate muscle contraction, and including electrodes like this one, which measures the transepithelial electrical resistance to measure how strong a barrier the cells have formed. However, most gut organ-on-chip systems are not designed with integrated sensors and still rely on offline or endpoint measurements, such as staining, in which basically you take a slice of the tissue, you kill the cells, you fix them, you break open their cell membranes and use antibodies to label specific parts of the cell. For instance, this is how we can see the location of serotonin, here labeled 5-HT. And that's how we see the spread of enterochromaffin cells and neurons, which contain the serotonin in the gut. But there's another way to detect serotonin, and that's by electrochemistry. This is one of the most versatile methods available. It's capable of sensing the molecule in real time as it's released from living cells with no chemical pretreatments or labeling needed. And it can be done using simple electronics and machinery. Thus far, the best serotonin measurement we have done is using dissected gut tissue, placing a carbon fiber electrode directly on top of the tissue, holding it at an oxidizing potential above the redox potential of the molecule. And every time serotonin is released from the tissue, we see a spike, just like that. So what we see is current being transferred from that oxidation reaction of the molecule at the electrode. So what we've been able to tell from these experiments is that the concentration of serotonin released from these tissues varies from less than 1 micromolar to over 10 micromolar or tens of micromolars. And that's quite a high concentration. Also, we can tell the timeline of how these spikes occur. And they can happen once every second or faster or slower depending on the kinds of stimulation. And what we see is that the different stimuli result in different patterns of serotonin release, which we know will affect the enteric nerves differently depending on the pattern that is released. However, there is a problem with these experiments. One, they're not only measuring serotonin. They're not only measuring, they're only measuring serotonin from the top side of the gut epithelium. As we saw on the previous slide, what we really care about is the serotonin that's released from the basolateral or bottom side of that layer. And that is exactly where those molecules will go on to stimulate the enteric nerves. However, it's difficult to get an electrode into the tissue and access those molecules, especially using real-time methods. What we have here is that gut-on-chip platforms provide a very convenient access to that bottom side of the epithelium. So our lab went to work using fabrication techniques to incorporate an electrochemical serotonin sensor on a cell culture platform. So here is the platform we've created. What we have is a 3D printed housing that contains a electrode integrated cell culture membrane. This is a porous membrane called polyethylene track etched or PET membrane. And on it, we have electrodes that we fabricated. So on the top side of the membrane, we have one type of electrode, which is for electrochemical impedance spectroscopy, or EIS, that measures physical coverage of the cells. Then on the bottom side of the membrane, we have an electrochemical electrode, which performs cyclic voltammetry to do molecular sensing, in this case, serotonin. So combine them, and we have multimodal detection capabilities on a chip that can grow cells directly on the actual sensor platform. So, for instance, EIS can be performed by applying a sinusoidal signal across the two electrodes, and the impedance or the capacitance of the solution will give you a certain readout. And when there's no cells cultured or low amounts of cells cultured on the membrane, you get one signal. And then as cells cover the membrane, that signal will change because the electrical properties of the surface have now changed. So what we actually see is that when there's no cells, impedance is relatively high. And as cells cover the membrane, we see the impedance decrease. And we think that that might be because the cells secrete conductive proteins that actually increase the conductivity of the local area of the electrodes. So another thing we can do is, in the future, stimulate the cells that are cultured there to then secrete serotonin. And since they're cultured on this porous membrane, the serotonin will diffuse directly through the membrane, and then we can use cyclic voltammetry to detect the serotonin in real time. So this is the ideal idea of how we see this platform being used. Thus far, we have characterized the sensors separately and without cells. So I'm just going to go through how we've characterized these sensors. But I'd also like to give a big shout out to Dr. Pradeep Rajasekaran. He was the originator of the idea of this platform, and he did all the impedance work. So thank you, Pradeep, for all of your help on that. So let's get into the serotonin detection scheme. Key is using this porous membrane, where ideally we would have cells cultured on the top, and we can stimulate the cells. For instance, butyrate is a short-chain fatty acid from bacteria. We would see serotonin secretion through the membrane, and we would see binding of that molecule to the electrode where it would be oxidized. How the signal works is that oxidation results in 2 electron transfer from the molecule itself to the electrode. So what we see is a current increase. And the reason that the serotonin is able to bind is we use this carbon nanotube structure to coat the electrode. And you'll notice that they share some structure similarity. Carbon nanotubes are made basically from rolled graphene, where they have these 6 structured benzene rings. And serotonin also has a benzene-like ring. So the theory is that they bind tightly together because of pi-pi stacking from these pi orbitals on the benzene structures. And so what we've shown is that indeed, by increasing volumes of carbon nanotube, you can increase the sensitivity of this electrode to serotonin compared to just a bare gold electrode on this membrane. And so let me just explain this curve. What we've done here is perform a CV, which is starting from a low voltage, you scan up to a higher voltage, which is above the redox potential of the molecule. And when you reach that redox potential, it catalyzes the oxidation of the molecule. And that's when those 2 electrons come and increase the current that's being measured at that voltage. So we can identify the molecule based on where on the scan it occurs, at what voltage, and the height of the current peak that occurs is proportional to the concentration of the molecule. So we've characterized how these electrodes work. So I'll go through how that works. One major point about this sensor is that the sensor is fabricated directly on the porous membrane. So this provides much better access to the actual molecules than if we used a downstream sensor. For instance, if you think of a microfluidic platform where cells are cultured in one section and you have to travel all the way down to another section of the platform to do the measurements, you've diluted the molecule quite a bit when you get to that second location. This distance between the cells and the sensors is only 15 microns, the thickness of the membrane. So what we've shown is that when using another kind of molecule, which is very standard and behaves in an ideal way for our tests, called ferrocene dimethanol, when we inject that molecule through the porous membrane, we see that although we expect final concentrations after full dilution in the whole platform to be on the micromolar range, we end up seeing it in the millimolar range because we are able to access 3 orders of magnitude higher concentration at that close site than it would be in the diluted bulk media. So that is a major advantage of putting sensors directly at the site of cell physiology. So let me illustrate another experiment we did, which also highlights the ability of these sensors to detect the dynamics of the sensing of the serotonin. So we did an experiment in lieu of having cells that actually secrete the serotonin. We did a sort of simulated experiment where we inject serotonin from a pipette and see how the sensor can monitor that over time. So if we start looking at just the baseline before an injection, and then we actually inject the molecule over some amount of time, the molecules will diffuse through the membrane, that oxidation will happen, and you'll get a peak. And then if you continue to just wait around and let the molecules diffuse further, you'll end up seeing exponential decrease in signal. So this follows a very standard and repeatable pattern, which sort of mimics how we would sense these molecular release events from the cells. And we're using cyclic voltammetry for this, which is not common in the literature. One reason for that is that it does have high sensitivity. So as I showed in my previous slide, cyclic voltammetry can very easily identify the concentration of the molecule, but it has relatively low time resolution. In this case, our sensors have minute to hour level measurement frequency. So these measurements are taken every 5 minutes, as we can tell, compared to the actual release events of every event from cells, which is on the second scale. This is longer than that. But our idea is that we can tune how these electrodes work. We can tune the amount of time that you actually need between every measurement to optimize the sensitivity, because the longer you let the electrode sit with that solution, the higher the sensitivity will be. So if we did want to have higher time resolution, another option is chronoamperometry, which does have that millisecond to second measurement frequency. Again, it has lower sensitivity, but higher time resolution. So this shows that we have options in terms of the time resolution and the concentration range that we want to detect from these cells. So let me talk a little bit about the future directions of this platform, especially for the multi-tissue monitoring that we'd really like to see. And of course, we work very closely, another co-PI of mine, Dr. William Bentley, and another collaborator, Jens Herberholz. They both contribute, and their labs contribute, the breadth of research that we need to bring this full device into the multi-tissue signaling domain. So what we also would love to incorporate is a module where we can incorporate actual bacteria from the gut microbiome, and we would culture them in mucus layer that is secreted from our model gut epithelium. Then we would also incorporate a model of the enteric nervous system, or even of the vagus nerve, which Jens, our close collaborator, does work in crayfish and does electrophysiology on the crayfish. So we would incorporate a crayfish nerve cord or gut itself in the bottom side to sense those serotonin molecules. So let me walk through a model experiment that we might do. So say we're interested in understanding the correlation between that Bacteroides fragilis bacteria and the downstream effects on the brain. We would introduce some of these pathogens. We would introduce some other stimuli that might trigger the serotonin release from these cells. And then we would see the serotonin both in our cyclic voltammetry or electrochemistry measurements, and we could see how that serotonin affects the downstream electrophysiology of the nerve cord itself. So you can see how this platform has the ability, depending on what kinds of gut cells you use. You could potentially use a biopsy from a patient to put their own cells and do a sort of diagnostic test on how their physiology is behaving. You could use cells from someone who's had a C-section in their early life to see if genetics plays a role, or if other components of the environment play the role, such as bacteria or medication or diet. And we could use serotonin as that clinical biomarker of both the gut disorders and potentially of certain brain disorders that are correlated with the gut signaling. So this sort of system can really advance gut-brain axis research of specifically understanding the molecular signaling and improve the clinical diagnostics by introducing serotonin as a biomarker of disease. So just to go through a couple more interesting future directions we're looking into, we have collaborators at Johns Hopkins who work with mice and humans as well, and are experienced in creating primary enteroids. So we would be able to use tissue from living animals, which is much more realistic than an immortalized cell line that many gut-on-chip systems are currently using. We also have a collaborator who does computational analysis, including cutting-edge image and video analysis. So we could evaluate, say, the motion and contraction of cells in correlation with serotonin and other stimulation, as well as using machine learning and prediction to, once the full system is designed, understand and predict how different forms and combinations of bacteria will actually affect the brain downstream. One big direction of our lab is to make ingestible capsules. We have this currently a prototype which senses trypsin as a enzyme that's produced from the pancreas as a biomarker of pancreatic diseases. But I'm interested in bringing our serotonin sensing capabilities to this sort of capsule using microneedles that would be present on the actual capsule so that it can get that basolateral intra-tissue serotonin measurements in as safe a way as possible. And then another direction we would love to look at, especially for treatments of gut diseases, is using smart probiotics, because bacteria have a home in the gut and they have the capability to respond to the signals that we can give them, including electrochemical signals. So in one theory that we are coming up with, we could detect serotonin patterns from epithelial cells, and depending on the pattern and the concentration, if it's in a range that is unhealthy, or we're suspecting that there's too much signaling going on, we can create another electrochemical signal, send it to the bacteria, and they would be genetically programmed to produce a drug that calms the effects and treats the patient. So with that, I would love to thank you for your time and ask for any further questions.
Yay. So yeah, always the weird part about these virtual Zoom ones. But really great talk. I really enjoyed some of your images as well. I thought it was like a really beautiful presentation also. So we have a couple questions so far. And so the first one is from Waldo Strange. And the question is, which bacteria were studied in the mouse anxiety trials you referenced earlier in your talk?
Well, they're putting me on the spot. I don't have, I can get back with you about exactly the specific bacterium. Generally in these studies, it's a variety. They look at the commensal bacteria, the ones that are expected to be in the gut, and they look at how they sort of vary. And so I think lactobacillus, different fragilis species are a couple of them, but I think I would have to actually refresh my memory on the specifics of those. Yeah, thank you for the question though.
Awesome. So Brantley Hall is just mentioning excellent talk. And then our president, Heidi Leonard, has a couple questions for you. And so I think when you were talking about cyclic photometry, you mentioned a little bit about time resolution. Is there a reason that you would need like high time resolution for this particular application?
Right. So this is, as I mentioned, like a very important factor when you're talking about real-time sensing. So what counts as real time, and why do we need it? So for instance, when we compare serotonin measurements to, say, ELISA, you know, an offline benchtop method that takes hours to do, and you get one data point out of it, and it takes handling and pre-treatment and human effort to get that data point. That really makes it hard to measure and monitor how molecules are changing in their concentration and their release over time. How fast you actually get that measurement, it's sort of up to the needs of the experiment. So as I mentioned, actually, I guess I can go back to this slide here. This is the main data that we have about serotonin release from gut tissue, and it shows that serotonin release is on the order of seconds. That's per release event. So you might say we need that 1 second time resolution to see exactly how the dynamics are changing at any given time. But our understanding is that really these conditions are chronic and they happen over really long periods of time. And it might still be effective to see the slowly, broadly changing concentrations from one day to another, or from one meal to after the meal, or you know, in correlation with things that take longer than just the second time scale of the actual individual release events. And also we're looking at multiple cells. A lot of these experiments, you sort of get your electrode close to one cell and you see release from there. But if we're looking at the average picture, I think, you know, we have minutes to hours time scale. I think hours is quite long for real-time sensing, but I think on the minutes time scale is effective. It would be effective, although we haven't officially actually measured these release events from our cultured cells yet because of culturing difficulties. But when we do that, we'll have to optimize what the time resolution should be for the stimuli that we see.
Cool. And then what's the specificity of cyclophotometry, or how can you make the sensor specific to 5-HT?
Right. So as you have seen, we use these carbon nanotubes on our sensors. So these are not particularly specific to serotonin. Technically, they can bind all sorts of different molecules. We do think that they enhance the binding of serotonin specifically, not in that they can't bind others, but serotonin is more enhanced in binding than, say, another molecule that doesn't do pi-pi stacking. So that's one measure of specificity. More so the specificity that we have is in the ability of CV itself to actually distinguish between different molecules that might come up in the solution. So if you had other redox molecules in the solution, which there might be in the body, of course, you would see peaks, but they might be at different locations on the CV scan. So you could still isolate the serotonin peak as long as they don't overlap too much.
Great. So I'm going to cut to a question from Ruben Acevedo. And so it's a two-part question. So basically, he's asking if you could elaborate a little bit more on the microneedle project. Specifically, would the needle act like a sensor, or are you injecting something into the gut?
Yeah, great question. We have a few projects going on actually with microneedles in our lab, and I've sort of tried to combine a couple of them. So for my vision of how this would work, we would actually use the microneedle as a sensor, so that the needle does two things. You can see we have some preliminary data showing that you can actually 3D print these needles using 2-photon polymerization, and you can print them with barbs that mimic how a parasite might stick itself into your gut. And so in one sense, you have a needle that can hold itself in the gut, ideally as safely as possible. And then using electrowetting and metal deposition, we can coat that needle with metal and with carbon nanotubes at the tip, so that that exposed tip can now be sensitive to serotonin measurements. And it would take some miniaturized electronics inside the capsule to do the CV and to do any downstream drug release or sending the signal out depending on what signal you get. But the needle itself would have the two functions of holding itself in the gut and also doing the serotonin measurement.
Great. So we had some two other comments. So Ebony Qualls and Jinjing Han said, "This is such great work and great job, Ashley," respectively. And then I guess our last question is going to be from Heidi. And the question is, how would you grow gut bacteria in a device like this? For example, how would you make sure that the environment is anaerobic while using this kind of sensor?
Yeah, really, really great question. And it's a difficult problem too. So we've talked about this quite a bit. It's not a trivial answer, but one option that we have is to grow the whole system in an anaerobic chamber and specifically feed the gut cells with oxygenated media, so that only those cells are actually getting the oxygen. So it would be similar to actually in your body, your gut, inside the actual gut is anaerobic because your body has absorbed all the oxygen before it gets to that point. So this section of it would be anaerobic, but on the bottom side, where your blood would be in the body, would have oxygen. So that's one option we're looking at. There's other options in terms of using bacteria that are not strict anaerobes, so that they can handle a little bit of oxygen. Some bacteria that are anaerobic completely die in the presence of oxygen, but we can choose the species that are going to be easier to work with to start off.
Great, awesome. All right, so I think we're going to end it there in the interest of time. But again, thank you so much, Ashley, for coming and talking about the gut-brain axis and everything. Thank you so much, and thank you everyone for tuning in. and we hope to see you all soon. Okay, bye everyone. Okay, I think we're good.